Okay, well, thank you very much for the invitation to speak at this wonderful conference. This is my first visit to ICRISAT, and I've really enjoyed it, especially given the fact that we have a half a meter of snow and minus 10 degrees centigrade temperatures at home. So this has been a welcome relief this week. Today I'm going to give you an overview of some of the work that we've been doing on optimizing training populations, integrating environmental covariates into prediction models, and some recent results on realized gain from genomic selection. Plant breeding is a predictive science. It is driven by new technologies and new knowledge, and everything that we do as plant breeders, we're trying to predict something. It might be performance in the farmer's field, it might be milling and baking quality, or some kind of disease resistance, but over time, breeders have developed methodologies for improving the heritability, improving gain from selection, and some of those things include family selection methods or progeny testing, but today we're interested in new strategies that take advantage of the power of using molecular markers. So the work that I'm going to talk about today mostly involves genomic selection. You've heard something about this at this conference, and I'm sure most of you have some familiarity with the methodology. It utilizes two different kinds of populations. The first population is the training population. This is a population of elite material that is genotyped with genome-wide markers. And we use those markers and we treat those as random effects and estimate their effects simultaneously in a single model. We assume that we have enough markers so that we have linkage disequilibrium between at least one marker and every QTL that affects the trait. Our prediction models attempt to capture the total additive genetic variation, and that allows us to estimate breeding values of individuals based on the sum of those effects or their kinship relationships. A second population we refer to as the breeding population. This population is genotyped with genome-wide markers, and we use that marker information in our prediction models to predict their breeding values in the absence of phenotypes. This figure just illustrates how those populations work together. The training population is composed of elite lines that are evaluated in your target population of environments, just as you would in your conventional breeding program. You use that phenotypic and genotypic information then to train your prediction model. That prediction model is then used in combination with either new germplasm or new populations coming out of your crossing program. That allows you to predict their breeding value, and then you choose the ones that have the highest breeding values to either recycle in your crossing program or move those into advanced trials. There are two essential approaches to implementing genomic selection. You can use biparental populations, or you can use multifamily populations, and they have different advantages and disadvantages. Biparental populations typically are used to train a model for each biparental population. So you use a portion of the population in your field evaluations, and you use that to train a model to predict the breeding value for the rest of the population. Obviously, this takes more time because you have to retrain the model for each population, but the advantage is that you don't have to worry so much about epistasis. You need fewer markers, smaller population sizes, and obviously you don't have to worry about minor allele frequency. It's really the only approach you can use for introgressing exotic germplasm. In multifamily approach, you build up a database over time and space to accumulate information that you can use to train new materials that are related to your multifamily training population. And this allows you to immediately generate breeding values instead of having to first train a model and then generate your predictions. This allows you to generate large training populations. You can use more genetic diversity, but typically you'll have lower prediction accuracy with this approach. This is a figure that just illustrates how I think you can integrate genomic selection into an applied breeding program. On the left is the rapid cycling stage, and this is intended to be able to take advantage of the fact that you don't have to phenotype every generation. So with a winter wheat population like what we use, you can plant and sprint, you can plant and harvest twice a year, and you can cross and self twice a year. From those self plants, that generates an F2 population, which then goes into the inbreeding stage, where you can then apply phenotypic selection or marker-assisted selection during the inbreeding stage. By the time you get to the F4 generation, you need to start thinking about a seed increase if you're going to then phenotype those selected lines. So at that stage, you have to generate, in the case of our wheat program, we have to have 100 grams minimum to put out a test plot. But at that stage, then you can, because you're predicting pure line performance with a pure line training population, you can then explore the possibility of capturing non-additive effects with models that have that capability. I'm going to tell you a little bit about some of the work that Nicolas Heslot has done. He's completed his PhD. He's back at Lima Grain in France. And when he came to Cornell, he said, I want to work on G/E. And I said, people have done that. There's nothing new to learn. Well, he proved us wrong. And I'll show you a couple of things in a couple of his projects. In this one, he wanted to use allele effects to identify outlier environments and identify relevant mega environments. And he used a Bayesian lasso, a common GS model. There was 1,000 barley lines, 58 environments, and it was an unbalanced data set with only about 18 genotypes present in more than half of the environments. So this is a typical breeding data set. In order to optimize the composition of the training population, he first started by taking each individual environment and building a prediction model for that environment, and then estimated its breeding values for all the other environments in order to get a mean accuracy for each individual training environment. Then he ranked them from low to high and removed one at a time the poorest prediction environment. He then retrained his model on the remaining environments and repeated the process. The environments that he took out of the predictive set, he moved into the unpredictive set, and then he ran the new model on the unpredictive set to see if some information was lost. This figure illustrates the results of that experiment. Up here is the predictive set. He started with 58 environments. Down here, the blue triangles are the unpredictive set, and the green squares are an independent validation set that came a year later. In the predictive set, he removed one environment at a time, and the prediction accuracy increased significantly up to 0.61, and then it began to decline again. When those environments were moved down in here into the unpredictive set, there was a slight increase in the prediction accuracy in the unpredictive set, but then it shot up at about the same place where the predictive set started to drop off. So that means that we were removing environments that had useful information for predicting the other environments. So the optimum set of environments was about 40 out of the 58. A second project that Nicola worked on was a project where he attempted to integrate environmental covariates in the prediction model to try to increase the prediction accuracy and to try to understand the underlying genetic architecture controlling the genotype by environment interaction. In this approach, he used a crop model, which you've heard about already today. So I won't need to get into what a crop model is. So the crop model they used was a model called Serious Quality. This is developed by a French group. And the first thing you have to do when you're using a crop model is you have to synchronize the maturity of the lines or the varieties that you have in your trial with the weather by calendar date, so that your developmental stages can be linked up at the appropriate time with each of the environmental covariates that you're using. The stress covariates were derived by developmental stage using information that we gathered from the literature about the sensitivity of different growth stages to different abiotic stresses. then these stress covariates were used as independent variables in a statistical genetic model. And this just shows some of the various stresses that occur at different stages of development in the wheat life cycle. So for the factorial regression model, each marker we fit a main effect and a sensitivity to each of the stress covariates. We had a total of 101 different stress covariates. There are different ways of measuring abiotic stress, and I'll show you an example in a minute. We also used a machine learning algorithm called soft rule fit for trying to capture the interactions between the markers and the covariates. So for each marker, so the performance was predicted as a main effect plus a G by E deviation. When you are using thousands of markers and 101 predictors, this becomes a computational challenge. So what we did, we reduced the dimensionality of the problem by first selecting markers that had a significant variance across the environments. And when we looked at the different markers for major genes, the only one that stood out was the photoperiod insensitivity gene called PPD-D1. However, that marker by itself did not explain a significant part of the G by E variation. In the end, our optimal model was based on 250 markers across the genome and a soft rule fit of level 4. So these are the most important stress covariates that ended up in our model. The most important one was the sum of the daily temperature from meiosis to flowering. And then there were 2 measures of early drought stress in the spring, and then follow that, 2 measures of heat stress before flowering, and then 2 measures of heat stress during early grain fill. So when you have a data set where you can predict genotype by environment interaction for any genotype in any environment, this allows you to build a 2-way table. And with those 2-way tables of lines by G by E, you can cluster those and investigate the relationships between environments and how they're producing this genotype by environment interaction. This is just a hierarchical agglomerative clustering of the environments involved in this experiment. There were 6 years, 5 years actually, and 44 environments in total. And you can see that in general, the environments within years were related to each other. So each year, the environments in each year grouped together, but overall there was a north-south trend in the data, and if you were able to sample more environments, you would probably be able to begin to group environments from different years in different locations. This figure shows the relationship between the accuracy when you included the G by E component and the accuracy without the G by E component. So the dots above the line represent improved accuracy when you include the G by E component. Overall, we obtained an 11% increase in the mean accuracy for the prediction and 11% decrease in the coefficient of variability. So using this approach, you can leverage your knowledge about agronomy, identify those stages of development in the crop where there's repeated stress, and try to develop germplasm and varieties that address those stress covariates. And it allows you to use existing data that you already have in your breeding program. Okay, so the next project that we set up was to investigate the importance of population structure in our genomic prediction accuracy. For this, we chose 5 different approaches to sampling subpopulations. That included random sampling, stratified sampling, the coefficient of determination mean, which maximizes the contrast between genotypes, the prediction error variance mean, which minimizes the error associated with predicting genotypic values, and then a combination of stratified sampling and coefficient of determination called strat CD mean. For this, we used two data sets, a wheat data set consisting of 1,000 genotypes and almost 39,000 GBS markers. For the rice, we had a population of 405 with almost 37,000 SNPs. That was an array-based genotyping platform. The traits for the wheat side were yield, test weight, lodging, heading date, and height. On the rice side, it was florets per panicle, flowering time, plant height, and protein content. For this experiment, we just used a standard GBLUP algorithm, and then the clustering was just using Ward's hierarchical clustering. In this figure, I want to illustrate how we went about sampling and evaluating the selected training populations. We start with a wheat data set, which we call the calibration set. Those are clustered and then sampled by stratified sampling. We mean that we selected individuals from each of the clusters in proportion to the size of the clusters, and then we generated training population sizes of 25, 50, 100, 200, and 300. Those, in each of those training populations were then used to predict the phenotypes and the breeding values for the test set. So that was a stratified sampling. Secondly, we used a CD mean directly on the calibration set, same with a PEV mean, and then we randomly sampled the calibration set. And then for the combination, we first clustered and then sampled within those clusters using the CD mean to generate our training populations. These are the two principal component axes for the wheat data set. We grouped them into 4 groups, and the first two axes explained about 20% of the variation. That we consider this to be a mild population structure. And it doesn't show up real well here, but these are the 3 PC analyses that shows what individuals were selected using the CD mean, the PEV mean, or the stratified CD mean. And you can probably see the red dots. Those are the ones that were selected most frequently out of the 50 runs. The green dots are the ones that were selected, and the black dots are the ones that were never selected by this algorithm. The CD mean tended to select individuals that were more in the middle and down here. The PEV mean tended to select individuals in this area. But the stratified CD mean was very effective at sampling the variation all across these PCs. In contrast, the rice data set showed extreme population structure, and about 40% of the variation was explained by the first two PC axes. For the sampling, this shows the CD mean, and again, the CD mean tended to group more in these individuals here. The PEV mean tended to sample more of these on this side, leaving out most of these. But the stratified CD mean was effective at selecting individuals throughout the range of the genetic variation that was explained by these PC axes. So these are the results of the sampling and the accuracy using different training population sizes. The training population sizes are shown along the bottom, and the accuracy is shown on the side. In the wheat data set, except for test weight and height, or test weight and heading date, the CD mean and the stratified CD mean were most effective. So those are the blue and the red lines. They're probably kind of difficult to see back there. But for test weight and heading date, the stratified and random sampling gave the highest accuracies. And I'm going to come back to that. In the rice data set, it was a little bit different picture here. The purple line shows the stratified sampling, and that pretty much gave us the best accuracy across all the population sizes. And recall that this was the population that had the most structure. But we wanted to try to understand why the CD mean did not perform as well for those two traits. And when we looked at the phenotypic variation captured by the CD mean, as shown along here, versus the phenotypic variation for the entire data set, what we found was that the prediction accuracy was reduced for these, and they captured less of the phenotypic variation compared to the other traits that performed better with the CD mean. We saw the same kind of a pattern in the rice data set, but the differences weren't as extreme. So it's important to remember that different algorithms you might have to explore different algorithms to be able to capture as much of that phenotypic variation as possible to maximize your accuracy. So the last project that I want to tell you about is a project that that was funded by the Bill and Melinda Gates Foundation. Jessa Rutkowski carried out the research to use genomic selection to select for adult plant resistance to the UG99 stem rust. This is a particularly virulent strain of stem rust, and it's overcome several genes in just a few years and spread from Uganda all the way up into Iran and down to South Africa. Jessica used a standard recurrent selection scheme where she intermated founders for 2 generations, and then self-individuals to produce S1 families. Those are phenotyped in the Njoro, Kenya stem rust screening nursery. And at the same time, those plants were also used for 2 cycles of genomic selection. We first trained our model using historical data. So this is data from that was used. This was data from the Njoro stem rust screening nursery over a period of 12 years, and we chose 373 individuals that had been evaluated in at least 4 environments, and the heritability is about 0.8 for those. We used 14 founders. Those were intermated for 2 generations to generate what we call our early generation selection candidate set. We divided that into 2 replicates of 252 individuals. And in the second year, we evaluated the C0 population in Kenya for one environment. In each cycle of selection for both the phenotype and the genomic selection, we selected 6 S1 individuals from 6 S1 progenies from 5 selected individuals, and those were intermated to create the next cycle of selection. For these populations, we used about 19,000 GBS markers, and that had less than 20% missing data. And those were imputed using expectation maximization. This is a PC analysis, again, showing first 2 principal components axes of the historical in black, the selection candidates in red, and the blue dots, the blue circles are the founder parents. When we looked at the prediction accuracies from the historical population and compared them to the early generation population, you can see right away that the early generation population, which had only been evaluated in 1 environment, gave us much better prediction accuracies. And this is clearly because they are more closely related to our selection candidates than the historical population. Another thing you can notice in this figure is that as the training population size increases, the prediction accuracy of both the early generation population and the historical population goes up. But whereas the historical population does not level off, you can see that the early generation population is already starting to level off. So this is characteristic of a situation where you have more close relationship, and with a smaller population size, you can get a better accuracy. These are the results from our genomic selection versus phenotypic selection for adult plant stem rust resistance. Remember, we only had time to do 1 cycle of phenotypic selection. We did 2 cycles of genomic selection, and we had 2 replicates. In Rep 1, shown on the top here, the phenotypic selection realized gain was greater than the genomic selection gain, whereas in Rep 2, it was just the opposite. Down here, the genomic selection realized gain was greater than the phenotypic selection. When we plot these on a graph showing the reduction in the severity of the rust versus the 2 years of selection, there was no significant difference, although the mean of the genomic selection severity was slightly less than that of the phenotypic selection. So, to summarize, genomic selection is complementary to marker-assisted selection and association breeding because it works best with quantitative traits of low heritability. It also does not require the knowledge of the underlying genetic control. You don't have to do a QTL analysis or the biological function, but you can use the products of genomic selection to try to understand the reason for some of the changes in the gene frequencies and the biological mechanisms. We found that integrating environmental covariates and crop modeling into the genomic selection framework increased our prediction accuracy and gave us some insight into how the genetic control of the genotype by environment interaction was contributing. And then finally, we found that genomic selection was effective for increasing quantitative stem rust resistance. This lists some of our funding sources. The Bill and Melinda Gates Foundation has been very generous. We also had grants from the U.S. Department of Agriculture. We're also extremely grateful to the Kenya Agriculture Research Institute for hosting the stem rust nursery and being such great hosts. We collaborate very closely with CIMMYT historically and in these projects, and it's been a wonderful relationship. And then finally, Lima Grain provided a fellowship for Nicola Heslow. I'll stop there, and I'd be glad to try to answer any questions you might have.
Was doing better even with 1 cycle of selection than 2 cycles of genotypic selection, which was converse in case of RAP2. Could you pinpoint what could be the reason?
Yeah.
To me, one could be that the heritability of resistance in this case must be pretty high, and you can straightway select for that.
I can give you a short answer right now, but I'd be glad to talk to you more about what we think actually happened a little bit later. First of all, the population sizes were quite small, so genetic drift could have been a factor. We weren't able to exactly test that with these populations, but that could be one factor. Also, if we were to do this again, I think we'd do 1 cycle of phenotypic selection before we started with genomic selection, and that's to generate some recombinants so that genomic selection can be more effective in its first cycle of selection. Also, if you notice the second cycle of genomic selection, the slope was greater, and that's because we trained the model based on that first C0 cycle of the selection candidates, and so that gave us higher prediction accuracies. I think those are the things that we think happened, and I think that in one of the replicates, the replicate where the phenotypic selection was more effective, we saw some unique recombinants. And we think that that gave the phenotypic selection an advantage in that replicate.
It's time for 2 more questions.
Leo from ACPFG, Australia. In the presence of population structure in your training population, how will it affect your prediction on the selection candidate? Is it a good thing or a bad thing?
Let me think if I can repeat the question. What is the impact of the population structure on genomic selection?
If you have population structure in your training population,
yeah.
How will it affect your prediction on your selection candidate?
When you're running a model on the entire training population instead of subpopulations, that prediction model could end up not being very predictive for some of the subpopulations. In that case, you may want to separate out those subpopulations and create a separate prediction model for those subpopulations where the prediction accuracy was inadequate. When you use a kinship model, GBLUP, in theory, you should be able to correct for that population structure in your prediction model. But when we look at that, we often see that some subpopulations do not have as good a prediction accuracy, and I don't understand why.